Alright, so this is the beginning of our topics associated with photosynthesis and light, and is covered in our textbook between chapters 6, 7, and 8. There is content that will be covered in chapters 13 and 14 on stresses and adaptations of plants as well. So that's kind of, mainly be looking at 6th, then 7th, and 8, but then bringing in those adaptations and stresses from 13 and 14 as we go through. So we'll start here with just a general introduction. This material here concentrates mainly on chapter 6. Oops, here we go. On chapter 6, associated with light and how light is used in photosynthesis. So the diagram that we're looking at is kind of a summary diagram that kind of looks back at this ecosystem level of all the components that are involved, all the different kinds of organisms involved in the movement of light energy through systems and the cycling of carbon within systems. So the organisms that basically capture solar energy using chlorophyll and other kinds of pigments are considered photoautotrophs, which means that they harness that energy using carbon as a source to harness that energy and make organic molecules, as you can see here in this part of the diagram, giving off oxygen as a byproduct. And now these organic molecules, such as glucose and sucrose and all kinds of organic molecules, can be used in cellular respiration by the plant or by a photoautotroph. During cellular respiration, the organism uses oxygen plus these organic molecules to make ATP, and then ATP can shuttle that harnessed energy that originally came from solar energy around to different parts of the cell to power different kinds of metabolic processes. So it's kind of our small change energy shuttling molecule. In the process of cellular respiration, carbon dioxide is given off plus water as byproducts, and then those, of course, are our reactants again to be used in photosynthesis. So basically plants are important, or photoautotrophs are important again for cycling carbon, and so carbon is always cycled within a system, and then of course harnessing energy. Whoops. There we go. Harnessing solar energy, which then moves through the system to some degree, but then is always lost here to some degree as heat. Now, as a whole system, including the heat loss, energy is never, it's always conserved, but it's basically transferring from solar to photoautotroph, and then we can add in where other organisms come in, such as heterotrophs, which are going to consume other organisms in order to acquire that carbon and energy. So here we go. We can actually insert the heterotrophs kind of in the middle here, rather than out here, where they are ingesting other organisms to acquire these organic molecules as well as the energy that is harnessed by those organic molecules, and then using oxygen also undergo cellular respiration to produce ATP, and in that process giving off carbon dioxide and water, which then is given up to the atmosphere and utilized once again by the photoautotrophs. So you can see how the heterotrophs kind of are included in this process here. Let's see. So we're going to concentrate on this aspect here, harnessing the solar energy by photoautotrophs, mainly concentrating on higher plants. So we'll go to the next page here. And just to kind of remind ourselves what our photosynthesis reaction is, in summary, not a balanced equation here by any means, but these are what we're looking for. using light energy. So carbon dioxide plus water using light energy will produce, in the very simplistic equation here, glucose, which helps us talk about respiration, cellular respiration, but really is a 3-carbon product initially that gets converted into all kinds of things. And so in terms of higher plants, this process, photosynthesis, is occurring at the leaf level, which you can see is green, because that's the color of the pigment chlorophyll. So we'll be talking a bit about chlorophyll. Light has to pass through the epidermis to reach these mesophyll cells. So the mesophyll cells, remember, contain the chlorophyll, or basically contain chloroplasts, which you can see kind of blown up over here. And each of these individual chloroplasts are found within the mesophyll. Here's a single one that's blown up to show the structures that you'll want to basically review. There's an outer and an inner membrane for the chloroplast rather. The fluid-filled space here is called the stroma, and then within the stroma are stacks of membranous sacs here called thylakoids. They're in stacks called grana, or a granum is a single stack. And the thylakoid membrane is where the chlorophyll molecules are embedded, and other pigment molecules are embedded in the thylakoid membrane, surrounding the thylakoid space, or lumen, which is what you'll hear us refer to it as, which helps compartmentalize various processes that are going on during the light reactions, basically this part of the reaction here, compartmentalizing the parts of the light reactions between the thylakoid membrane and the stroma. And then of course so we could separate the two processes of photosynthesis as the light reactions, which, just to write down here, occur within the on the thylakoid membranes. Plus the thylakoid lumen, plus the stroma. So there are things happening on either side of those thylakoid membranes. And this is where water and light are utilized as reactants, which produce ATP, as we'll see, plus NADPH. ATP harnessing the energy, NADPH collecting electrons from chlorophyll that are energized there. All right, then the second phase is referred to as the Calvin cycle, or the PCR cycle. And this is where CO2 is going to be utilized. So we can see up in the figure here, carbon dioxide comes into the leaf through stomata and will diffuse in through the different spaces surrounding the mesophyll cells, the leaf air spaces, the substomatal spaces, towards the mesophyll cells. And basically CO2 has to diffuse across these membranes to where it's utilized within the stroma. So the Calvin cycle occurs in the stroma, and it is a series of intermediates that are produced in the process of converting carbon dioxide to a 3-carbon sugar. All right, so the products of the Calvin cycle are sugars, and then the recycling of ADP plus inorganic phosphate and NADP plus to capture electrons. Now we can, we'll be talking about this shortly, but we can revisit some themes here associated with this reaction, where there's a, this is essentially a reduction oxidation reaction, where carbon dioxide is gaining electrons here and so therefore is reduced, and water is donating electrons or losing electrons and becoming oxygen, so water is oxidized. So we'll come back to talking about what gets reduced and what gets oxidized. Also, real quick, just to emphasize, you do need to be familiar with the various labels and different kinds of cells that make up the leaf, where carbon dioxide is coming in, oxygen is coming out through stomata, how water is actually getting to the leaves and diffusing across these membranes into the thylakoid spaces, where that water is going to become important in the light reactions. So make sure you can identify all the parts of the leaf and the parts of the chloroplast, as well as where chlorophyll would be found, and in what parts of the chloroplast these different reactions or parts of photosynthesis are taking place. Okay, so let's take a look at light now. And here we're looking at a diagram of how light is represented. Basically, light has a few details here we want to take note of. It's basically made of discrete packets of energy called photons. And photons will propagate through space in waves. Depending on the amount of energy contained within the photons determines the wavelength of the light, basically the difference between, or the distance between the two crests here determines the wavelength. So as we said, light propagates through space in waves, and it's measured, energy is measured or is determined by wavelengths, which is the distance from crest to crest. Okay, and that distance determines, that wavelength in other words, determines the energy contained in a photon of light from within, that's propagating through space. So we're going to take a look here now at light within the electromagnetic spectrum, which is the whole range of energy that light can contain, a whole range of wavelengths that are determine the amount of energy that light contains. So we can see at the smallest end of the range, in terms of the smallest wavelengths, is gamma rays, so 10 to the negative fifth to 10 to the negative third nanometers of light, or of wavelengths basically, which contain the highest energy. And then at the opposite end we have the long wave radiation, which has the lowest energy. And long wave radiation has wavelengths on the order of 1,000 meters or kilometers, anywhere from 1 to maybe 10 to 1,000 kilometers. So the longer the wavelength, the lower the energy. The analogy we used in class was if you throw a pebble into a lake, the initial impact creates the shortest wavelengths of ripples, which is where the energy is highest in quantity. And then as the ripples move away from where the rock entered the pond, the wavelengths increase and therefore they contain less energy. So that's kind of an equivalent to how, or an analogy to the way we can look at light. The part of the part of the electromagnetic spectrum that we're interested in is, for plants, is the visible spectrum, which ranges from 380 nanometers to 750 nanometers. So the 380 nanometers occurs in the region of the visible spectrum that we usually associate with indigo, violet, and blue. 750, with its lower amount of energy, occurs in the red region. All right, so the energy of a photon is determined by the wavelength, which is symbolized as lambda here, of light that photon comes from. For example, the energy associated with light from the blue indigo region would be approximately 274 kilojoules, whereas the energy associated at the opposite end of the visible spectrum in the red region is contains about 181 kilojoules of energy. So we're interested in concentrating on that visible spectrum from the violet blue to the red region from 380 to 750 nanometers.